Hello, and thank you for joining me on this gentle journey. Tonight, we will slowly drift into calmness and explore the quiet waters where the axolotl lives, suspended in a world that feels untouched by time. In the dim, green glow of ancient canals, this small creature moves like a soft shadow beneath the surface, drifting without hurry or resistance. The axolotl carries a quiet wonder within it, the ability to regenerate parts of its body, as if life itself can gently repair what was once lost. Let your thoughts soften as you imagine this peaceful underwater world where everything moves slowly and nothing needs to be rushed. If you enjoy this relaxing moment, please consider liking, subscribing, or sharing your thoughts below. But for now, simply breathe and relax. Let's begin. In the quiet southern reaches of Mexico City, the waterways of Xochimilco stretch like faded memories of an ancient inland sea, a landscape where water and earth refuse to fully separate. Here one sees narrow canals threading through floating gardens, known as Chinampas, their edges woven with reeds and roots that seem to hold the land together against slow-motion currents. Light filters downward through the still air in muted greens and browns, as if filtered through layers of time itself, rather than water alone. It is within this subdued, sediment-rich world that the axolotl drifts, a living presence moving through submerged roots and drifting vegetation with unhurried precision. Unlike the rapid ecosystems found in more turbulent environments, the canals maintain a slow ecological rhythm that has persisted for centuries, allowing species like the axolotl to remain closely tied to ancient biological patterns. Sediment settles gently along the canal floors, creating a soft, almost prehistoric substrate where light barely penetrates and movement becomes more a matter of vibration than vision. In this environment, survival is not dictated by speed or dominance, but by adaptation to stillness, by the ability to interpret subtle shifts in water and shadow. The canals are not merely habitat, they are a preserved fragment of Earth's biological memory, where evolutionary time appears to slow almost to a pause. For the axolotl, this stability is not a limitation, but a defining condition of existence, one that has shaped its physiology over thousands of years without requiring drastic change. It moves through the canals with minimal effort, conserving energy in a way that reflects the scarcity of necessity within such a stable ecosystem. Every drift through the water is slow and deliberate, as if the surrounding environment has removed the need for urgency entirely. In such stillness, evolution itself appears to have paused, leaving behind a creature that embodies continuity rather than transformation. The canals, fed by remnants of ancient lake systems that once covered the Valley of Mexico, carry within them layers of ecological history that extend far beyond human settlement. Within these waters, organic matter accumulates slowly, forming a rich but unstable foundation for life that depends on balance rather than abundance. The axolotl drifts through submerged roots, its movements barely disturbing the suspended particles that drift like dust in an underwater atmosphere. In this dim environment, perception is shaped not by clarity, but by sensitivity to change, however slight. Here, the axolotl does not compete in the conventional sense of predation. Instead, it occupies a quiet niche defined by patience and timing. It waits motionless among aquatic plants, relying on environmental cues that signal the presence of small organisms drifting within reach. Feeding, when it occurs, is sudden and precise, a brief interruption in an otherwise continuous state of stillness. Between these moments, life returns to near silence as currents carry microscopic life forms through the canals in slow circulation. The water itself acts as both medium and memory, preserving traces of movement long after disturbances have passed. Each ripple carries information about nearby activity, translating physical motion into a language the axolotl's sensory systems can interpret. Over time, this creates an environment where survival is less about confrontation and more about awareness. In such conditions, energy is conserved not as a strategy, but as a necessity embedded within the ecosystem itself. The surrounding landscape, shaped by centuries of human cultivation and natural sedimentation, continues to shift slowly beneath layers of ecological time. Even as modern development presses outward, fragments of these ancient waters remain, holding onto a version of the world that predates the city's expansion. The axolotl moves through this fragile continuity, neither rushing toward change nor resisting it, but existing entirely within its steady rhythm. In this way, it becomes a quiet expression of biological persistence, a form of life that endures by remaining still as the world transforms around it. Within the shallow, plant-filled waters of central Mexico, the axolotl presents one of the most unusual developmental patterns known among vertebrates. Rather than undergoing full metamorphosis like most amphibians, it remains in a juvenile form throughout its entire life, retaining external gills, a finned tail, and an entirely aquatic existence. This condition is known as Neoteny, a biological state in which reproductive capability is reached without the typical transition into a terrestrial adult stage. Biologists studying amphibian development first recognized this phenomenon as more than a simple variation in growth. In most salamanders, hormonal signals, particularly those involving thyroid activity, initiate a dramatic transformation. Limbs strengthen, lungs become dominant, and aquatic structures recede. In the axolotl, however, these signals are naturally suppressed or altered, allowing juvenile characteristics to persist indefinitely. This deviation is not a defect in development, but a stable evolutionary outcome shaped by environmental conditions. Deep within the canals, where water temperatures remain relatively stable across seasons, the pressure to adapt to land-based environments has historically been minimal. In such surroundings, remaining aquatic offers continuous access to food sources and shelter without the energetic demands associated with metamorphosis. Over generations, natural selection favored individuals that retained larval traits, reinforcing a life cycle centered entirely within water. From a physiological standpoint, the axolotl's endocrine system regulates growth in a markedly different way compared to other amphibians. Thyroid hormones, which normally trigger metamorphic change, are present, but do not activate the full cascade of developmental shifts. As a result, gills remain external and functional, allowing efficient oxygen exchange directly from the surrounding water. This adaptation enables the species to remain submerged without reliance on atmospheric breathing structures. Remarkably, this permanent juvenile state also influences metabolism. Energy expenditure remains relatively low, supporting a lifestyle built around slow movement and infrequent bursts of activity. Instead of allocating resources towards structural transformation, the organism directs energy toward maintenance, survival, and reproduction within its aquatic niche. The result is a life history strategy that prioritizes stability over transformation. Genetic studies have revealed that this trait is deeply embedded in the species' regulatory networks. Specific genes responsible for metamorphic signaling are either downregulated or expressed differently, creating a biological framework that supports long-term larval morphology. Researchers examining these pathways have noted that small changes in hormonal sensitivity can dramatically alter developmental outcomes, demonstrating how finely tuned vertebrate growth systems can be. In laboratory environments, the axolotl has provided valuable insight into developmental biology. Its transparent juvenile features allow scientists to observe organ formation and tissue maintenance without invasive techniques. Yet, despite its prominence in research, the same biological traits that make it scientifically valuable are rooted in an ecological history tied specifically to the waters of Xochimilco. Behaviorally, this organism exhibits patterns consistent with a fully aquatic existence. Feeding, resting, and reproduction all occur without transition to land. External gills, resembling delicate feathered structures, continuously move to facilitate respiration, extracting oxygen directly from the water column. These features, maintained throughout life, illustrate how neoteny is not simply arrested development, but a complete alternative life strategy. Across evolutionary time, this adaptation reflects a broader principle. Change is not always necessary for survival when environmental conditions remain stable. In the case of the axolotl, remaining in a juvenile state has proven more successful than undergoing transformation. Its existence demonstrates how developmental flexibility can lead to entirely different evolutionary pathways, shaped by subtle interactions between genetics, hormones, and environment. Within the still waters of, this organism continues to inhabit a form that predates many of the ecological pressures seen in other amphibians, maintaining a biological identity that is both specialized and remarkably consistent across generations. Among vertebrates, few organisms demonstrate restorative capacity as extensive as the axolotl, a species capable of rebuilding entire limbs, sections of spinal cord, portions of the heart, and even regions of the brain with high structural accuracy. In the waterways, this ability is not an occasional biological anomaly, but a consistent survival feature shaped by evolutionary time and repeated exposure to injury within a complex aquatic environment. Following tissue loss, the initial response in the axolotl involves rapid wound closure without scar formation. Skin cells migrate across the damaged surface, forming a protective layer that stabilizes the area, while deeper regenerative mechanisms activate beneath it. Unlike mammals, where fibrosis often replaces functional tissue with structural scar material, the axolotl maintains a regenerative pathway that preserves the original architecture of the injured region. Cellular activity soon intensifies beneath the wound site, where mature cells near the injury boundary revert to a more flexible, stem-like state. This transformation leads to the formation of a structure known as a blastema, a mass of proliferating cells that serves as the foundation for new tissue growth. Within this structure, cells remain highly responsive to biochemical signals that guide their eventual specialization into muscle, bone, nerve, or connective tissue. Genetic regulation plays a central role in directing this reconstruction. Developmental genes typically active during embryonic growth are reactivated in a controlled sequence, allowing the organism to rebuild structures that closely match the original form. Research has identified signaling pathways involving fibroblast growth factors, proteins, and other molecular regulators that coordinate this regeneration with remarkable precision. These pathways ensure that newly formed tissues integrate seamlessly with existing structures. In aquatic habitats, injury risks arise from predators, environmental debris, and fluctuating water conditions. The ability to restore lost appendages provides a significant survival advantage, reducing the long-term consequences of physical damage. A regenerated limb, once fully developed, regains full functionality, including musculature, skeletal alignment, and neural connectivity. Neurological regeneration in the axolotl is particularly notable. When portions of the spinal cord are damaged, neural cells can regrow across the affected region, re-establishing communication pathways between the brain and body. This restoration occurs without the permanent loss of motor function that is typical in most vertebrates. The capacity for neural reconnection suggests a level of plasticity that extends beyond simple tissue replacement. At the molecular level, injury triggers a cascade of signals that reprogram local cells. Instead of forming scar tissue, fibroblasts and other structural cells adopt a more embryonic-like state. This reversion enables them to respond dynamically to positional information within the body, ensuring that regrown structures align correctly with pre-existing anatomy. Spatial patterning is preserved through gradients of signaling molecules that guide growth direction and tissue differentiation. Laboratory studies have shown that this regenerative ability remains consistent even under controlled conditions, where environmental variables are minimized. Researchers observe that limb regrowth follows a predictable sequence: wound stabilization, blastema formation, rapid cellular proliferation, and gradual differentiation into specialized tissues. Each stage is tightly regulated, minimizing errors in reconstruction. From an evolutionary perspective, such extensive regeneration likely developed as a response to persistent ecological pressures in ancient lake systems. In environments where predation and physical damage were common, the ability to restore lost body parts increased survival probability significantly. Over time, natural selection reinforced genetic pathways that support this capability, embedding it deeply within the species' biology. In broader scientific research, the axolotl serves as a model for understanding regenerative medicine. Its biological systems provide insight into how vertebrate tissues might be encouraged to repair themselves more effectively, offering potential applications in treating spinal injuries, organ damage, and degenerative diseases in other species. Within its native waters, this organism continues to rely on a form of biological restoration that replaces loss with renewed structure, maintaining continuity of form through mechanisms that remain among the most sophisticated observed in vertebrate life. Within the sediment-heavy canals, visibility often collapses into a narrow range of just a few centimeters, where suspended particles absorb and scatter incoming light. In such an environment, the axolotl operates without reliance on sharp vision. Instead, depending on a combination of mechanosensory and chemical detection systems that translate water movement into actionable information. Extending along the sides of its body, a specialized sensory network known as the lateral line system allows the axolotl to detect minute changes in water displacement. These receptors respond to pressure waves generated by nearby movement, enabling the organism to identify prey, predators, or obstacles even in complete visual obscurity. This system is similar in function to that found in fish, reflecting deep evolutionary connections among aquatic vertebrates. When small organisms such as insect larvae or tiny crustaceans move through the water column, they create subtle vibrations that travel outward in all directions. The axolotl interprets these signals as spatial information, constructing a functional map of its surroundings without needing direct sight. This form of perception transforms the surrounding water into an active medium filled with continuous sensory input. Chemical detection also plays a significant role in environmental awareness. Dissolved compounds released by potential prey or decaying organic matter disperse through the canal system, forming invisible gradients that the axolotl can sample through specialized receptors. These olfactory cues help it distinguish between edible targets and inedible material, refining its feeding decisions in a habitat where visual confirmation is often unreliable. Feeding behavior is closely aligned with these sensory adaptations. Instead of pursuing prey over long distances, the axolotl remains stationary for extended periods, conserving energy while monitoring environmental signals. Once a suitable target enters its sensory range, it executes a rapid suction-based strike, drawing both water and prey into its mouth in a fraction of a second. This motion depends on the sudden expansion of the buccal cavity, generating a localized pressure difference that pulls material inward. In the canals, where plant matter and organic debris frequently cloud the water, this hunting strategy proves highly effective. The inability to rely on visual tracking is compensated by heightened sensitivity to movement and chemical traces, allowing the axolotl to function efficiently in conditions that would challenge many other aquatic predators. Neural integration of these sensory inputs occurs in the brain, where signals from the lateral line system and chemical receptors are combined into a coherent spatial representation. Although the axolotl's brain is relatively simple compared to that of mammals, it is sufficiently specialized to interpret complex environmental cues and coordinate precise motor responses. This integration enables accurate timing during prey capture, even when targets are not directly visible. The aquatic environment itself amplifies the importance of vibration-based perception. Water transmits mechanical disturbances more effectively than air, allowing information to travel rapidly across short distances. Every movement within the canal system contributes to a constantly shifting field of sensory data, shaping how the axolotl perceives its surroundings moment by moment. Predatory interactions in this environment are defined less by pursuit and more by detection and timing. The axolotl's success depends on its ability to distinguish meaningful signals from background noise, a task that requires continuous sensory filtering. This selective awareness ensures that energy is expended only when necessary, aligning with its overall low-energy survival strategy. Even in the presence of near-total visual limitation, the organism maintains a stable orientation within its habitat. Spatial awareness is reinforced through repeated exposure to consistent environmental structures such as submerged roots, sediment layers, and slow-moving currents. These features provide reference points that assist in navigation without requiring visual confirmation. Over evolutionary time, this sensory specialization has allowed the axolotl to thrive in habitats where clarity is rare and unpredictability is constant. Its perception is shaped not by sight alone, but by an intricate combination of mechanical and chemical awareness that transforms opaque water into a detailed sensory landscape. Across the layered history of central Mexico, the axolotl occupies a rare position where biological reality and cultural meaning intersect. Long before modern scientific classification, it was already embedded in the worldview of the Mexica civilization, where it was associated with transformation, water, and the boundary between life and death. Its name is derived from Xolotl, a deity linked to movement between forms and states of existence, reflecting how this amphibian was interpreted as something suspended between worlds. Within the ancient lake basin that once dominated the Valley of Mexico, the axolotl thrived in an extensive network of freshwater systems. Over centuries, those lakes were gradually reduced through urban expansion, agricultural drainage, and the growth of modern infrastructure. What remains today in Xochimilco is a fragmented remnant of that aquatic landscape, sustained through artificial channels and Chinampa agriculture, where floating gardens still rest upon stabilized sediment beds. In these remaining waterways, the axolotl now exists under significant ecological pressure. Water quality has declined due to urban runoff, nutrient imbalance, and contamination from surrounding development. Elevated levels of pollution alter oxygen availability and disrupt the delicate balance required for sensitive aquatic life. At the same time, introduced species such as tilapia and carp compete for resources and directly prey upon axolotl eggs and juveniles, reducing reproductive success in the wild. Despite these challenges, the species continues to persist in isolated pockets of suitable habitat. Conservation efforts have focused on restoring water quality through filtration systems, reintroducing native vegetation, and constructing refuge zones designed to limit exposure to invasive predators. These interventions aim to recreate conditions that resemble the ecological stability of earlier centuries, though achieving full restoration remains complex. Scientific interest in the axolotl has also contributed to its survival in indirect ways. Laboratory breeding programs across multiple countries maintain genetically diverse populations, ensuring that the species is preserved outside its natural environment. These controlled populations are used for research in developmental biology and regenerative medicine, yet they also function as genetic reservoirs that may support future reintroduction efforts if natural habitats recover sufficiently. Cultural perception continues to influence conservation priorities. In modern Mexico, the axolotl has become a symbol of ecological vulnerability and national identity, appearing in education campaigns, public art, and environmental awareness programs. This symbolic presence contrasts sharply with its declining wild populations, highlighting the tension between cultural recognition and ecological reality. Historically, the Chinampa system played a critical role in maintaining aquatic biodiversity. These artificial islands, built from layered sediment and vegetation, created productive agricultural zones while supporting complex freshwater ecosystems. Over time, however, changes in water management and urban expansion reduced the ecological function of these systems, contributing to habitat fragmentation for native species, including the axolotl. Genetic studies reveal that the wild population has undergone significant reduction in both size and distribution. This decline increases vulnerability to environmental fluctuations and reduces long-term genetic diversity. Conservation biologists emphasize that maintaining even small, stable populations in natural conditions is essential for preserving evolutionary potential. Within this narrowing ecological space, the axolotl continues to reproduce through external fertilization, releasing eggs into submerged vegetation where they develop independently. Success rates in the wild are now heavily dependent on localized water conditions, predator presence, and habitat stability. Efforts to restore ecological balance in these canals involve collaboration between scientists, local communities, and environmental organizations. Water management strategies aim to improve circulation and reduce contamination, while habitat reconstruction seeks to rebuild submerged plant structures that support breeding and shelter. Even as its natural range contracts, the axolotl remains deeply intertwined with the landscape, representing both a surviving fragment of ancient aquatic ecosystems and a living indicator of environmental change. Its continued existence reflects the fragile equilibrium between human activity and biological persistence in one of the most altered freshwater systems in North America. Deep within the soft-bodied structure of the axolotl lies a nervous system that challenges long-held assumptions about vertebrate limitations. In the freshwater channels, this organism survives not only through external regeneration of limbs, but also through an extraordinary capacity to restore internal neural networks after damage. Neuroscientists studying spinal and brain injuries in vertebrates have long observed that most species respond to neural trauma with scar formation and permanent loss of function. In contrast, the axolotl demonstrates a markedly different biological response. When neural tissue is damaged, surrounding cells activate developmental pathways typically reserved for embryonic stages, enabling reconstruction of functional neural connections rather than permanent structural blockage. Following injury to the spinal cord, specialized glial cells in the axolotl do not form dense inhibitory scar tissue. Instead, they support regrowth by creating a permissive environment for axonal extension. This allows nerve fibers to extend across damaged regions and reconnect with their original targets. Motor and sensory functions can be restored with a level of completeness rarely observed in other vertebrates. At the molecular level, this capability is linked to the reactivation of genes associated with early development. These genes regulate cellular identity, guiding mature cells to temporarily adopt more flexible states. Once in this state, they contribute to rebuilding neural pathways by responding to chemical gradients and positional cues within surrounding tissue. Research conducted on the axolotl has identified signaling molecules such as fibroblast growth factors and transforming growth factor pathways as key regulators of neural restoration. These molecular signals coordinate cell migration, differentiation, and structural alignment, ensuring that regenerated tissue integrates correctly with existing neural architecture. In aquatic environments like those found in Xochimilco, injury from predators or environmental hazards can disrupt both peripheral limbs and central nervous structures. The ability to restore neural connectivity provides a critical survival advantage, allowing individuals to regain mobility and sensory coordination even after severe physical trauma. Laboratory observations reveal that newly formed neural tissue in axolotls closely mirrors the organization of original structures. Axons extend along defined pathways, guided by extracellular matrix components that provide spatial orientation. This level of precision suggests that the organism retains a form of biological memory encoded not only in surviving neurons, but also in surrounding supportive tissues. Unlike mammals, where adult neural plasticity is limited, the axolotl maintains a persistent capacity for structural adaptation within its nervous system. Synaptic connections can be re-established following disruption, and functional circuits can be reconstructed without the extensive compensatory rewiring typically required in other species. Developmental biologists have noted that this regenerative capacity is closely tied to the species' broader life history traits. Because the axolotl retains juvenile characteristics throughout life, many of its tissues remain in a state that is more receptive to growth and reorganization. This neotenic condition contributes to the persistence of regenerative pathways that are normally suppressed in adult stages of other vertebrates. In experimental settings, axolotls have demonstrated the ability to recover coordinated movement following partial spinal transection. Over time, motor function returns as neural pathways re-establish communication between brain and musculature. The restored system is not merely a partial replacement, but often a highly functional reconstruction of original circuitry. Further investigation into this organism has revealed that its nervous system operates with a high degree of plasticity, even in uninjured states. Synaptic remodeling occurs throughout life, allowing continuous adaptation to environmental changes. This flexibility supports both regeneration and ongoing refinement of neural connections. Within the ecological context, such resilience ensures survival in a habitat where physical disruption is common. Predation events, environmental fluctuations, and human-induced changes all contribute to conditions where recovery of neural function can determine long-term viability. Across vertebrate biology, the axolotl stands as one of the most significant models for understanding how nervous systems can rebuild themselves after severe injury. Its ability to restore internal wiring offers a rare example of functional recovery that extends beyond compensation into true structural renewal. In the submerged channels it inhabits, this organism continues to navigate its environment, using a nervous system capable of rebuilding itself from within, maintaining continuity of behavior and movement, even after events that would permanently impair most other vertebrates. Movement across the submerged vegetation often appears minimal at first glance, yet within that stillness, the axolotl engages in a highly specialized method of feeding, shaped by sensory precision and energy conservation. Rather than pursuing prey through active chase, it relies on detection, timing, and a rapid mechanical response that defines its role as an ambush predator in low-visibility waters. Suspended particles and decomposing plant matter fill the canals, limiting long-range visual tracking and making pursuit inefficient. In response, the axolotl has evolved a strategy centered on stillness, paired with heightened environmental awareness. It positions itself among roots and submerged vegetation, where water flow is reduced and passing organisms are more likely to drift within range. Small aquatic organisms such as insect larvae, worms, and crustaceans move through the water column in irregular patterns, generating faint hydrodynamic disturbances. The axolotl detects these signals through mechanosensory systems along its body, allowing it to interpret direction and distance without relying on sight. This sensory input is integrated with chemical cues, providing additional information about potential food sources. When a target enters an optimal range, the feeding response is triggered almost instantaneously. The axolotl expands its buccal cavity in a rapid motion, creating a sudden pressure drop that draws water and prey inward. This suction-based capture mechanism is highly efficient, requiring minimal physical displacement while maximizing intake success. The entire action occurs in a fraction of a second, leaving little opportunity for prey escape. Hydrodynamic efficiency plays a central role in this feeding behavior. By minimizing forward motion and relying on internal pressure changes, the axolotl reduces water disturbance that might otherwise alert nearby organisms. This allows repeated feeding attempts within the same location without significantly altering the surrounding environment. Energy conservation is a defining feature of this strategy. Extended periods of inactivity between feeding events reduce metabolic expenditure, aligning with the relatively low energy demands of its aquatic habitat. In the nutrient-variable waters, such efficiency increases survival probability during periods when prey density fluctuates. The structure of the axolotl's skull and jaw supports this specialized feeding technique. Musculature associated with buccal expansion is capable of rapid contraction, while skeletal elements remain flexible enough to accommodate sudden volume changes within the oral cavity. These anatomical features work in coordination to generate controlled suction without compromising structural stability. Sensory feedback continues even during feeding events. Mechanoreceptors detect changes in water flow as prey is drawn inward, allowing the organism to adjust strike timing and orientation in real time. This ensures that capture remains accurate even when environmental conditions shift unpredictably. Feeding frequency is influenced by both environmental availability and internal metabolic state. In habitats where prey density is low, the axolotl can sustain long intervals without food intake, relying on its ability to reduce energy consumption during inactivity. When conditions improve, feeding activity increases proportionally, demonstrating flexible behavioral adaptation. Within the canal ecosystems, this feeding strategy positions the axolotl as a mid-level aquatic predator, interacting primarily with small invertebrate populations. Its role contributes to maintaining balance within micro-ecosystems, where shifts in population dynamics can influence broader ecological stability. Comparative studies with other amphibians show that this suction-based feeding mechanism is particularly effective in low-visibility aquatic environments. Unlike visual hunters that depend on clarity and pursuit, the axolotl operates through proximity-based detection, allowing it to function effectively in sediment-rich water systems. The coordination of sensory input, muscular response, and skeletal flexibility reflects a finely tuned evolutionary adaptation. Each feeding event represents a brief convergence of multiple biological systems working in unison, resulting in a capture method that is both rapid and controlled. Across its life cycle, the axolotl maintains this feeding strategy without significant alteration, demonstrating its long-term effectiveness within stable aquatic environments. Its survival is closely tied to this balance between stillness and sudden action, where minimal movement is paired with precise mechanical execution. Through the long ecological history of the Valley of Mexico, the axolotl represents an evolutionary path that diverges from the typical amphibian trajectory. While most members of its broader taxonomic group transition between aquatic and terrestrial life stages, this species remains permanently within water, retaining larval characteristics throughout adulthood. Paleontological and genetic evidence suggests that ancestral populations of axolotls originated in ancient lake systems that once covered the basin surrounding present-day Xochimilco. These lakes provided stable aquatic environments with minimal seasonal disruption, reducing the selective pressure for land-based adaptation. In such conditions, traits associated with terrestrial mobility offered little survival advantage. Over extended periods, natural selection favored individuals that maintained efficient aquatic respiration and morphology. External gills, streamlined body structure, and finned tails remained advantageous for navigating still, vegetation-rich waters. As a result, the species retained juvenile anatomical features while reaching reproductive maturity, establishing a stable neotenic life cycle. At the hormonal level, this developmental stability is primarily linked to regulation of thyroid activity. In most amphibians, thyroid hormones initiate metamorphosis, triggering structural and physiological changes necessary for terrestrial life. In the axolotl, these hormonal signals are naturally modulated in a way that prevents full activation of metamorphic pathways, allowing larval features to persist indefinitely. Genetic studies have identified variations in hormone sensitivity and receptor activity that contribute to this outcome. Rather than a complete absence of developmental triggers, the system exhibits altered responsiveness, resulting in a life history strategy centered entirely on aquatic existence. This represents a form of evolutionary specialization rather than incomplete development. The ecological stability of ancient lake environments played a central role in reinforcing this adaptation. Within the waters that once dominated the region of Xochimilco, food resources, shelter, and reproductive sites were consistently available in aquatic form. Under these conditions, terrestrial exploration did not significantly increase survival or reproductive success. Morphological traits associated with aquatic life became increasingly refined over time. The axolotl's limbs remain externally structured for movement along submerged surfaces rather than weight-bearing locomotion on land. Its respiratory system continues to rely on external gill structures, which extract oxygen directly from water with high efficiency in low oxygen conditions. Comparative analysis with closely related salamander species demonstrates that metamorphosis remains a potential developmental pathway under certain environmental triggers. Laboratory experiments have shown that changes in water availability, hormonal manipulation, or environmental stress can induce partial or complete metamorphosis in axolotls. However, such transformations are not typical in natural populations, where stable aquatic conditions persist. In evolutionary terms, this species illustrates that adaptation does not always follow a linear progression toward increased terrestrial complexity. Instead, it demonstrates that specialization within a stable ecological niche can be equally successful. By remaining within aquatic environments, the axolotl avoids the energetic and structural costs associated with metamorphosis. Within the fragmented ecosystems of modern Xochimilco, this evolutionary strategy now faces new challenges. Habitat alteration and environmental disruption introduce conditions that differ significantly from those under which its traits originally evolved. Despite these changes, the species continues to express the same developmental pattern, highlighting the persistence of deeply embedded genetic regulation. Research into this organism continues to inform broader understanding of vertebrate evolution, Its life history provides a clear example of how environmental stability can shape developmental pathways over long timescales, resulting in organisms that remain closely tied to ancestral ecological conditions. The axolotl therefore stands as a living example of evolutionary continuity, maintaining an aquatic form that reflects both ancient lake ecosystems and the long-term influence of hormonal and genetic regulation within a stable environmental niche. Inside controlled research facilities across the world, the axolotl has become one of the most intensively studied amphibians in modern biology. Its unusual combination of developmental stability, regenerative ability, and aquatic specialization has placed it at the center of investigations in genetics, developmental biology, and regenerative medicine. Across institutions, axolotls are maintained in carefully regulated aquatic environments that replicate aspects of their native habitat in Xochimilco. These laboratory populations allow researchers to observe growth, reproduction, and tissue development under stable and repeatable conditions. Such environments provide clarity that is difficult to achieve in the increasingly altered natural canals where wild populations persist. Embryonic transparency is one of the most valuable characteristics for scientific observation. Early-stage development can be monitored directly without invasive procedures, allowing researchers to track organ formation, neural development, and limb growth in real time. This visibility has made the axolotl a key model for studying vertebrate development from its earliest stages. Regenerative biology remains the primary area of focus. Scientists investigate how the axolotl reconstructs complex structures, such as limbs, spinal tissue, and portions of internal organs. By examining gene expression during regeneration, researchers identify molecular pathways that regulate cellular identity, differentiation, and spatial organization. These pathways include signaling networks that guide cells to rebuild structures with high anatomical accuracy. In parallel, genetic sequencing has provided deeper insight into the regulatory architecture of this species. Comparisons between axolotls and other salamanders reveal differences in gene activation patterns, particularly in pathways related to hormonal response and tissue repair. These differences help explain why regenerative capacity and neotenic development remain active throughout the axolotl's life cycle. Despite its prominence in research environments, the species continues to exist in fragile natural conditions. In the canals of Xochimilco, wild populations face habitat degradation, water contamination, and competition from invasive species. This contrast between laboratory abundance and environmental scarcity highlights a growing divide between controlled study populations and natural ecosystems. Laboratory maintenance of axolotls ensures long-term genetic preservation, but it does not replicate the full ecological complexity of their native environment. Variables such as fluctuating water chemistry, natural vegetation structure, and ecological interactions are difficult to reproduce outside of their original habitat. As a result, wild populations remain essential for understanding the species in its complete ecological context. Scientific interest in the axolotl extends beyond basic biology into potential medical applications. Research into its regenerative abilities informs studies on tissue engineering, wound healing, and nervous system recovery. By analyzing how this organism restores damaged structures, scientists aim to identify principles that could guide future therapeutic approaches in other vertebrates. Ethical considerations also accompany its widespread use in research. Conservation biology emphasizes the importance of maintaining natural populations alongside laboratory colonies, ensuring that scientific study does not replace ecological preservation. This dual responsibility reflects the species' position at the intersection of science and environmental conservation. Within this dual existence, natural habitat and laboratory environment, the axolotl continues to serve as both a subject of scientific inquiry and a living component of a diminishing ecosystem. Its role in research underscores the connection between biological discovery and the urgent need to protect the environments from which such knowledge originates. Along the remaining waterways, the final strongholds of the axolotl persist within a landscape that has been reshaped repeatedly by human activity. These canals, once part of an extensive lake system, now exist as fragmented ecological remnants surrounded by one of the largest urban centers in the Western Hemisphere. Urban expansion has gradually altered water flow, sediment composition, and nutrient balance across the region. Agricultural runoff, wastewater input, and infrastructural development have introduced conditions that differ significantly from those in which the species originally evolved. These changes have affected oxygen availability and overall water quality, placing additional stress on remaining wild populations. In response to these environmental pressures, conservation programs have focused on restoring portions of the canal system through habitat reconstruction and water treatment initiatives. Floating vegetation platforms, filtration channels, and controlled breeding zones have been introduced in an effort to stabilize ecological conditions. These interventions aim to recreate elements of the original aquatic environment while maintaining compatibility with surrounding human infrastructure. In parallel, captive breeding efforts continue in research institutions and conservation facilities worldwide. Genetic management strategies are used to preserve diversity within laboratory populations, ensuring that long-term survival remains possible even if wild numbers continue to decline. These populations also serve as potential sources for future reintroduction, provided that habitat conditions can support sustainable survival. Within this changing environment, the axolotl maintains its characteristic biological stability. Its life cycle remains centered entirely in water, with external gills and larval morphology preserved throughout adulthood. Reproduction continues through egg deposition in submerged vegetation, although success rates in the wild are increasingly dependent on localized environmental conditions. Ecological surveys indicate that wild populations have become highly restricted in distribution, occupying only a fraction of their historical range. This contraction reflects broader trends in freshwater biodiversity loss associated with urbanization and habitat fragmentation. Despite these reductions, small populations persist in isolated sections of the canal network where conditions remain suitable. Cultural recognition of the species has grown alongside scientific interest. In modern Mexico, the axolotl appears in educational programs, public art, and conservation messaging as an emblem of ecological fragility and biological uniqueness. This visibility has contributed to increased awareness of freshwater ecosystem preservation. Across scientific, literature, and environmental monitoring reports, the axolotl is frequently cited as an indicator species, reflecting the health of its aquatic habitat. Changes in its population size and distribution are used to assess broader ecological stability within the Xochimilco system. Even as its natural environment continues to transform, the axolotl endures in a form largely unchanged by external pressures. Its continued existence represents a biological continuity linked to ancient lake ecosystems that once defined central Mexico's geography. Within these remaining waters, the species persists as both a biological remnant and a living measure of environmental change, reflecting the long-term consequences of ecological disruption and the ongoing efforts required to maintain fragile aquatic systems. Its continued presence depends on a delicate balance between restoration initiatives and the pressures of surrounding urban growth, where even small shifts in water quality can influence survival. As monitoring efforts expand and habitat management becomes more refined, scientists and conservationists use the axolotl as a living indicator of ecosystem health, tracking changes that are often invisible at the surface. In this way, its survival is closely tied to broader environmental stability, where each improvement or decline in the canal system echoes through the structure of the species itself, shaping its future in ways that remain deeply connected to the fate of its shrinking habitat. As our journey through the quiet waters of the axolotl comes to a close, picture the still canals fading into darkness and the gentle creature drifting peacefully below the surface, unchanged and unhurried. There is a quiet lesson in its world. Life does not always need to transform quickly. Sometimes it simply continues, softly and steadily. Let the gentle sensations of peace and calmness settle through your body and mind. Feel your thoughts slow down, your breathing deepen, and your body become lighter and more relaxed with each passing moment. Rest here in this quiet stillness, fully supported in calm. If you enjoyed this relaxing moment, please consider liking, subscribing, or sharing your thoughts below. You can also turn on the notification so you don't miss any of our future journeys together. But for now, let your body relax, release all tension, and allow your mind to drift gently into rest. Good night and sleep well.